All right, good afternoon, everyone. Thank you again for joining us today for our last grand rounds for this academic year. We have our special guest, Dr. Theoharides, who's our professor and vice chair of clinical immunology. He's also the director of the Institute for Neuroimmune Medicine at Clearwater and the executive director for the Center of Excellence for Neuroinflammation Research here at KPCOM. He is also an adjunct professor of immunology at Tufts School of Medicine, where he was professor and director of molecular immunopharmacology and drug discovery and clinical pharmacologist, Massachusetts Drug Formulary Commission. He served on the board of the Institute of Pharmaceutical Research and Technology and the Supreme Health Council of the Ministry of Health in Greece. He received his BA, MS, MPHIL, PhD, and MD degrees and the Winsternitz Prize, I'm sorry if I said that incorrectly, in pathology from Yale University. He also received a certificate in global leadership from Tufts Fletcher School of Law, in diplomacy, and a fellowship at Harvard Kennedy School of Government. He trained in internal medicine at New England Medical Center, which awarded him the Oliver Smith Award, recognizing excellence, compassion, and service. He received the Tufts Distinguished Faculty Recognition and Excellence in Teaching Awards multiple times. He showed that the unique tissue immune cells, mast cells, communicate with the brain microglia and are critical for neuroinflammation. He has 500 scientific publications, which equates to 50,000 citations with an H index 114, placing him in the world's top 0.05% of most cited authors and the worldwide expert on mast cells by Scholar GPS and ExpertScape. He was inducted into the Alpha Omega Alpha National Medical Honor Society, the Rare Disease Hall of Fame, and the World Academy of Sciences. He developed novel dietary flavonoid supplements formulated in olive pumice oil to increase absorption. He has received 37 patents and trademarks. And with that, I hand it over to Dr. Theoharides.
Thank you very much. Well, today we'll talk about a topic that I've been studying for 40 years. So don't mind me if I'm very passionate about it. So our educational objectives will be to cover the mast cell biology as quickly as we can, focus on brain mast cells, the blood-brain barrier, and microglia activation, describe some processes involved in long COVID, and I interjected actually some information about myalgic encephalomyelitis, chronic fatigue syndrome. And then we'll finish, if we have enough time, with some potential ways of regulating mast cell activation. I am the scientific director of a company called Algonaut that makes dietary supplements, but I will not actually talk about any of those supplements today. So first of all, let's talk about mast cells. You may remember from either histology or pathology that they're everywhere in the tissues, as we will discuss. But what are the diseases we primarily talk about? So we have the so-called atopic disorders, and atopic disorder is actually a subclass of allergies because they're typically only associated with immunoglobulin E. And those are the bread and butter of what we see every day: allergic conjunctivitis, allergic rhinitis, asthma, anaphylaxis. But then we have the non-atopic disorders that do not involve IgE, even though we throw them under the class of allergies. Such would be hereditary angioedema, mastocytosis, urticaria pigmentosa, etc. And if anybody is interested, there's, I think, a wonderful video on YouTube that I helped made with two actual patients, one from Denmark, one from the United States, called My Mystery Symptoms and Mast Cells, that encompasses a lot of the things, or most of the things, we will discuss today. So many times patients ask me, well, why don't you write something in a journal that everybody reads in the scientific community? Because who reads allergy journals? So it took us about 5 years and about 40 drafts before we had actually this paper published in the New England Journal of Medicine about 10 years ago. And I was grateful to the journal because they allowed us, for the title, to use mast cells, mastocytosis, which is a rare condition, but also related disorders, which opens up the field to what we will discuss today. So before we look at the right-hand side boxes, let's look at the left-hand side. We divide mast cell disorders into 3 categories. So primary, such as mastocytosis, which is rare, occurs in 1 in 20,000 people or so. And these individuals have a lot of mast cells everywhere in the body, which happen to also be superactivated. Then we have the secondary class, which is pretty much what we consider allergies, as I mentioned earlier, such as urticarias, sometimes inflammation around solid tumors, etc. And then we have idiopathic, which means we don't understand what on earth is going on. And we have idiopathic anaphylaxis, angioedema, itching, and this condition, mast cell activation syndrome, that everybody, it seems, talks about these days. But I will try to distinguish from what is listed there as mast cell activation unspecified, because we don't necessarily have to be hung up with MCAS. Now, the right-hand side shows a lot of the confusing aspects of dealing with mast cell disorders. First of all, as you see, there are many diagnostic codes, and you can choose and pick, basically. So if someone of your patient does not necessarily qualify for mast cell activation syndrome, which I will describe in detail shortly, you can take your pick. So you can have secondary mast cell activation. You can have mast cell activation unspecified. So you can always make the patient feel better by saying, yes, you have SATs, because sometimes they need to know that they have SATs to feel better. But as we go down the list, there are also conditions that are very confusing, such as multiple chemical sensitivity. And we don't have a specific diagnostic code for that. If you look towards the bottom, sometimes we use toxic effects, sometimes we use unspecified respiratory conditions, sometimes we say unspecified allergies. So on the one hand, they're very confusing, but on the other, it allows you to use diagnostic codes for your patients. So don't despair if they don't fit a particular one that maybe they wish they did. But if you look at the bottom box, we also make our lives very difficult in medicine because we use terms that are very poorly described or identified and overlapping. So we say allergies, we say atopic disorders, as I already said, mast cell disease or disorders, mast cell activation disorders, mast cell activation associated disorders, and the list goes on. So we are, as physicians, as providers, also at fault by not sticking to a few descriptions that make life easier for everybody. Unfortunately, this is what we have to live with. Now, how about the mast cells? First of all, historically, an experiment was done in 1901 out of the Oceanographic Museum of the Principality of Monaco. And I happened to give a lecture there, so the Prince of Monaco gave me a copy of this slide. So two scientists, Portier and Richet, were going to actually ground up tentacles of jellyfish, were going to inject them into dogs, and they were certain the dogs were going to be actually immune to any subsequent stings. And they had a word ready for it. They were going to call that phenomenon prophylaxis. Instead, when the dogs were exposed to the stings, they actually constricted the bronchi, dropped their blood pressure, and they died. So they called it anaphylaxis. But no one knew what that meant. Now, at about the same time that you see in the center panel in 1887, Dr. Paul Ehrlich, who got the Nobel Prize at that time, were staining certain tissues and used a dye called toluidine blue. And what do you know? Some cells turned violet. And you can see at the left-hand side an intact, compact cell, and on the right-hand side a cell that is more sort of expanded. He didn't know what it was all about, but you could identify those punctate violet dots, and he thought that this cell was feeding surrounding tissues. So you use the Greek word for breast, mastos, like mastectomy, and you called it mastocytes or mast cells, except that these cells don't feed other cells, and he was wrong naming them, but the name stuck. So these cells are made in the bone marrow as precursor cells. They do not circulate as mature cells that go into the tissues, and they mature under various different circumstances or local microenvironment. And what we used to think one mast cell is one mast cell, it doesn't hold water anymore. Mast cells are very different in different tissues. But they contain about 1,000 of these little dots, secretory granules. And as we will see, each one of those granules contains about 50 molecules. Now, uniquely, they're found around blood vessels. If you look at the right-hand side at the bottom, you see a cross-section of a blood vessel, and the violet cells shown by the red arrows are mast cells. So mast cells are found in all tissues, especially those exposed to the outside, such as eyes, nose, lungs, skin, gut. But if you look at the left-hand side at the bottom, this wonderful paper showed mast cells in the bifurcation of a blood vessel shown as red. The mast cell is the sort of green-blue. And the arrowhead indicates that the mast cell can throw philopodia, tentacles, between the endothelial cells that make up the blood vessel, and they sense what's happening in the blood vessel as well. So the mast cells are the very first cells that will sense danger, whether it's outside the body or inside the blood vessels, and they will act as a conductor and orchestrate an immune response, about which we will have plenty to say. Now, in a nutshell, if we look at the caricature of the mast cell, you see at about 11 o'clock the receptors for immunoglobulin E, and these are the typical receptors for allergens. If they're bridged by an allergen or antigen, there's a flux of calcium ions that leads eventually to what is called degranulation. And if you look at the left-hand side at the bottom from our laboratory back in Boston, the brown is the nucleus. The very tight blue dark granules are intact granules, and the light sort of blown up granules, or the content of the granules, is what has been released. So this is a very quick reaction, as I will show you with the last slide if we get to it. And molecules that are pre-stored in the granules, such as heparin, histamine, platelet activating factor, proteolytic enzymes like tryptase, get released immediately. Now, at about 7 to 10 years or so, another receptor was identified, shown at about 9 o'clock, called MRGPRX2. This is a low affinity receptor, meaning it needs a lot of a stimulus to trigger the cells, and it's mostly triggered by positively charged drugs and peptides. But what is unique and shown at the left-hand side in this publication is that this receptor talks to the receptor for immunoglobulin E. So one is activated, the other is actually ready to fire. So one feeds the other. Now, what is amazing about the cells, and we've been studying for years, is what happens to the right-hand side. And of course, the receptors are all over the cell, not to one side or the other. But at about 1 o'clock, you see high affinity receptors for neuropeptides released from nerves. At about, 2 o'clock or so, you see receptors for cytokines, especially interleukin-1 and interleukin-33. I'll tell you more about these. And then at about 3 o'clock or so, you see receptors, TLR, toll-like receptors, activated by bacteria, fungi, and viruses. And in fact, a wonderful paper was published by my colleague, Dr. Afrin and Moldering, shown in the middle panel there, where they identified about 350 different molecules that can be released from mast cells, yet we mostly deal with histamine and maybe leukotrienes. And what is amazing about the cell is that when they're triggered by this activation of the right-hand side sort of receptors, there's no degranulation. The cell looks perfectly happy at you on light microscopy, but it releases molecules shown at the bottom box. So the first bullet is inflammatory cytokines, the second bullet inflammatory chemokines, and the third bullet inflammatory peptides. And there is tons of evidence that the mast cells communicate with nerves, which I will say repeatedly during this presentation. At the left-hand side, you see the cover of Journal of Investigative Dermatology. The red is a blood vessel. The wiggly lines are actually nerve endings, sensor nerve endings, and the greenish are actually cells from mast cells. And the mast cell has a lot of molecules that are both itching-causing, pruritogenic, as well as nociceptive, such as adenosine, bradykinin, histamine, platelet activator, etc. And what the evidence shows more and more is that this new receptor, the MRG receptor, can be activated by peptides, especially substance P. Then the mast cell releases molecules shown in the middle caricature that activates the nerves, and then you have a cyclic reaction going. And more papers indicate that the mast cell is also involved in pain, although I will not have time to discuss this today. But just to make the whole life more complicated is that if you look at the right-hand side, this was created in the publication that you see at the top left about 2 years or so ago, and you see how many additional receptors are actually found on the surface of the mast cell. And such receptors can be triggered, as you can see in the caricature in the middle, by drugs, by peptides, by cytokines, by environmental triggers. And if you look at the middle panel, you see even more receptors being identified, every year. And two very important type of receptors that we will discuss a little later again are the ones shown at the bottom. These are the TRPV4 receptors that are involved actually in stimulation by various chemicals, as well as aryl hydrocarbon receptors also involved in stimulation of the mast cells by various chemicals that places the mast cell at a very critical position for chemical sensitivity syndrome. But the mast cell, as I said, does not necessarily degranulate. So I'll take credit. I published this paper as a graduate student at Yale in 1978. And the picture on the right-hand side is from the journal Science, where we published that paper. And we cause the mast cells to release either individual granules or many granules or many, many more granules, which means that every mast cell does not necessarily respond the same way at any particular time. And we're right now with my colleagues in the laboratory at Davy, we're trying to figure out why some cells, as you can see at the top, do not respond, and some cells at the bottom do respond, hoping that we might be able to identify with RNA sequencing what might be present in the mast cells that don't respond that might lead us to treatment. But if you look at the caricature at the left-hand side, you see that the mast cell can, of course, undergo the degranulation that I mentioned. It can go through something called compound exocytosis, piecemeal degranulation, differential release. It can actually release extracellular vesicles or exosomes that our colleague Lubov actually studying for other cell types. And it create actually nanotubules. It can even make synapses. So our thinking that the mast cell has to degranulate to participate in disease, it's really absolutely wrong. We're only looking at the tip of the iceberg. And we and others have written reviews such as the one shown at the bottom about differential release of mast cells and how that might participate in inflammation. You might ask, if it doesn't degranulate, how does it do it? So here's a paper published with one of my graduate students from Yale, back in 2003, where we stimulated the human culture mast cells with interleukin-1, and only interleukin-6 was released. And if you look at the right-hand side, this is a cryo-immunoelectron microscopy, again, from my lab back at Yale. And what we see is a cluster of little dots being released from part of a mast cell. So we're looking at about, tenth of a mast cell. And each one of those dots is an interleukin-6 particle recognized at electron microscopy because it is bound by an antibody, which is bound to a gold particle, which is actually obviously detected at electron microscopy. So you might say, well, what is the big deal? Well, the big deal is that this cluster is as small as a synaptic vesicle releasing neurotransmitters. It's about 1/20th the size of the secretory granules that contain histamine. And you might then ask, well, why is interleukin-6 important? Why don't we measure it? Well, we should be measuring it because you can see at least 3 papers independently shown that interleukin-6 reflects the severity of mastocytosis in many individuals. Another colleague showed at the bottom that interleukin-6 promotes actually proliferation of mast cells. And at the right-hand side, if the mast cells have a mutation shown there, they consistently make interleukin-6, and that causes proliferation of mast cells. So the fact that we say, well, interleukin-6 is made by so many other cells, why bother measure it, is outright wrong. We should be measuring it because along with other indices, it tells us a lot about what the mast cells are doing and the symptoms of the patient. And then we went ahead with graduate students of mine again at Yale, and we published 3 papers in the Proceedings of the National Academy of Sciences, where we showed that if we stimulated the human mast cell with substance P, a peptide released from nerve endings, along with the cytokine interleukin-3, we see thousands of units of TNF, hundreds of units of interleukin-1, and again, hundreds of units of vascular endothelial growth factor, which is also vasodilatory. Well, I don't know of any cell type in the immune system that can release so much TNF, IL-1 beta, or VEGF than mast cells, yet we still don't consider the mast cells as part of the immediate immune response. We only think of it in terms of allergies. And as I said earlier, the mast cells communicate with other cells. They communicate with T cells. They communicate with M macrophages. They have toll-like receptors shown in the red box. And I just chose 3 publications to make the point to the right-hand side. So you can see Borrelia, that actually causes Lyme disease, can stimulate mast cells to release only cytokines. Sporothrix, a type of fungus, can release interleukin-6 TNF, again with no degranulation. And this paper at the bottom shows that influenza A virus can stimulate the mast cells without degranulation to release cytokines. So the evidence is there if we really want to look at it. But what becomes even more complicated is that the mast cell responds to heavy metals, like to mercury, to aluminum. They respond to herbicides such as atrazine and glyphosate, which we all know is Roundup, and in my life I don't know why it's still allowed to be sold. And at the bottom you see metal ions stimulate the mast cells, as well as chemicals such as malathion. And with a colleague from BU, Dr. Sullivan, we're trying to actually figure out if Gulf War illness patients who had a lot of chemical sensitivities might actually have succumbed to that because of mast cell activation. But I'll kind of throw my social self out there to indicate that there are tremendous hypersensitivity reactions that involve mast cells to excipients in drugs. And many drugs don't tell us about the excipients. If you look at the bottom, you see early life exposure for children to endocrine disruptive chemicals that can affect actually neurodevelopment in children. And at the right-hand side, you see neurotoxic effects of propionic acid. Propionic acid is made by bacteria in the gut, but it is the most popular preservative in foods, supplements, and cosmetics. Go figure. And I'm glad that the New England Journal of Medicine wrote this editorial about manufactured chemicals and children's health, the need for a new law to somehow limit this, and we're not quite there yet. So, summarizing view is, the mast cells as shown in the middle can be activated by different triggers that can release different molecules, and depending on the molecules released, that can affect every organ in the body. So not just skin and itching. And many patients come, especially to us at the clinic, having symptoms that affect like every organ in the body, and they've been through as many physicians in the past in as many years, and everybody told them, you're crazy. You can't have it all. And yet, yes, they can. And it gets even more complicated if you look at the table at the right-hand side, because many conditions mimic mast cell-related issues. They might not be necessarily due to mast cells, but they're very confusing. So, for instance, carcinoid syndrome releases primarily serotonin, not histamine, but the symptoms are almost identical to histamine release. You've got diarrhea, you've got flushing, you've got hypotension, you've got headaches, etc. So it is very challenging to identify, diagnose, and deal with patients that have multiple problems. So if I were to give you an algorithm, which is taken from the New England Journal of Medicine article, if someone has telltale signs of mast cell activation, and those might be a reaction to foods, medications, contrast media, etc., you've got to work them up. So either you measure this proteolytic enzyme tryptase, and it's got certain cutoff limits, such as more than 20 nanograms per milliliter for mastocytosis. I'll get into this shortly. Or you have these lesions on the skin that look like hives, but they itch all the time. And mostly when you scratch them, actually, that's called a derriere sign. But you have those, you almost don't have to do a biopsy. You can do a skin biopsy, and that's called cutaneous mastocytosis. Or you can do flow cytometry that we are trying to do now in the lab as well for certain receptors found on mast cells. If they're present, then literally the patient will buy a bone marrow biopsy. And in the bone marrow biopsy, as you see at the bottom, you see islands of 50 or more mast cells, not just 2 or 3. And if you don't see those, then you do urine measurements from breakdown products of mast cell mediators, such as methylhistamine, etc. And if those are high, we call that sort of mast cell activation syndrome. Now, mast cell activation syndrome was identified by colleagues Peter Vallen, Metcalf, and Akin some years back. And it was interesting, if you see the blue box, they specifically said accompanied by neurologic complaints. Yet over the years, the neurologic complaints have dropped off. Even though my good friend Larry Afrin published at least this paper saying an appreciated cause of neurologic and psychiatric symptoms for mast cell activation. And Dr. Butterfield later published numerous papers that if we were to measure in the urine, 24-hour urine, now we say even first morning void might be sufficient, metabolite of prostaglandin or leukotriene, which is not shown there, that might be even better than measuring serum tryptase. What is interesting, and I don't have time to go into it, is what is shown at the bottom publications, where mast cells seem to be somehow more prevalent in patients that also have Ehlers-Danlos syndrome, which is a hypermobile syndrome, as well as dysautonomia, especially POTS, which is postural orthostatic tachycardia syndrome. We don't know why those go together, and sometimes we call them a triad. Now, Dr. Lyons, many years ago, also identified a genetic condition where instead of the beta-2 tetramer of tryptase found inside the granules, showing at the top sort of red granule, these are alpha-2 dimers through a genetic condition, but the alpha-2 dimers are not in the granules. They are released. We don't know how they're released, but if you look at the left-hand side, the symptoms are very similar to an allergic reaction. So you've got pain, headache, flushing, itching, irritable bowel-like syndrome, etc. And we have no idea why, because the alpha-2 dimer is inactive as compared to the tetramer of the tryptase, which is active. So questions is, how is it released? How can we inhibit it? How can it kind of lead to all those symptoms? But since then, there have been a lot of changes which don't filter into all of us that deal with patients, such as, for instance, for the normal level, it used to be 11 nanograms per milliliter, now it's gone up to 15. And you have to actually measure within 48 hours of an episode. So unless someone has an indwelling catheter, it's almost impossible to measure tryptase. And because of that, my friend and colleague Mariana Castells from the Brigham at Harvard wrote this editorial about elevated tryptase conditions and pitfalls, and it was based primarily on the publication shown at the bottom, where they looked basically at high tryptase levels over 15 nanograms per milliliter or so. And if you look at the red box, they identified that most of them were associated with hypertryptisemia or, God forbid, chronic kidney disease, and then some leukemias. So that was the first we had ever heard that tryptase might be elevated actually in chronic kidney disease. And colleagues of mine from Greece and I wrote a review last year about the role of mast cells in chronic kidney diseases. And then we also have to deal with a new syndrome that is constantly being recognized called alpha-gal. Alpha-gal is an antigen found in mammals, but we somehow don't, it doesn't bother us. But if we're bitten by a tick, regardless whether we get Lyme disease or not, the saliva of the tick has bitten actually a squirrel or a deer or a cow and took basically the alpha-gal with it. When it bites a human, now it makes human exquisitely sensitive to alpha-gal, which is measured, by the way, by LabCorp and Quest. And the amazing thing, again, is that the symptoms could be even within a few minutes or days to weeks later. And as you can see, it's not just itching. It could be coughing. It could be dropping blood pressure. It could be nausea, vomiting. You can see how confusing this can be when a patient presents. And finally, regardless of mast cell activation, we have what is called histamine intolerance. So there's a lot of histamine in many foods. And you can see the green box: avocado, cheese, nectarines, sardines, peanuts, spices, tomato, tuna, all of them have it. And if the individuals have enzymes that's supposed to break down histamine, and the enzymes have polymorphisms or mutations, they cannot tolerate histamine. And you can see at the bottom, COMT, CYP3, diminoxidase are enzymes that break down histamine. And why is that important? Because as you see at the left-hand side, it was published in the journal Nature some years back. If in fact someone responds to food, then if you look at the caricature from an editorial in the New England Journal of Medicine, histamine and other molecules sensitize sensory nerve endings in the gut, and that registers as abdominal pain in the brain. That might be what we actually have in children that have colicky pain, and as it turns out, they're probably allergic or sensitive to cow milk products. So how do we diagnose these conditions? Well, if you look at the patient and assuming they haven't, slept for 3 days, which might cause the problem, if you see black circles under the eyes, we call them allergic signers. That's sort of a telltale sign we might be dealing with allergic problems. And if you scratch someone's back or the underarm, and in 2 minutes you get this red line, it's called dermatographia, that means the mast cells are ready to fire just to the pressure. There's no allergen there. Now, if you look at the laboratory tests, and I'm not going to go through all of them, we have the ones that are called mandatory, such as measuring immunoglobulin E and tryptase, and the ones in 24-hour urine or first morning void, but the urine must be kept and sent to the lab cold. If it warms up even 15 minutes at room temperature, all the measurements are gone. And then there are additional recommended tests depending what we suspect. So we have a lot of itching, we do different tests. If there's a lot of edema, different. If there's inflammation, different, so forth and so on. So we don't necessarily have to ask for all of them. But on the other hand, if someone presents with 20 different symptoms, we might have to cover the waterfront and measure all of them. And as I said, we can measure the 24-hour urine, and it's important to do genetic polymorphism because of this mutation shown at the very first that is associated with muscle proliferation, diamine oxidase, methyltransferase. Those are the two enzymes that break down histamine. And in chronic spontaneous urticaria, meaning it occurs every now and then, you itch like crazy, about 50% of the people don't respond to antihistamines at all. And that's when we do the basophil activation test. It comes with different names, but basically there's autoantibodies against either the IgE or the IgG receptor, and the basophils are circulating, and they have this receptor just like the mast cells do, so we can actually stimulate the basophils with the patient's plasma, and if histamine is released, that indicates that there is actually the presence of autoimmune antibodies. Clearly, it would be better if we had mast cells, and you can see at the bottom mast cells versus basophil activation test. And again, with my colleagues in the lab, we're trying to set up actually a mast cell activation test, which would be available for anywhere in the world, basically. But I said earlier that the muscles communicate with neurons. And at the right-hand side, you see a paper published many years ago, where the top shows the vertical line is a blood vessel, the smaller line, the wiggly line, is a sensory nerve ending, and there's a mast cell piggybacking there. And if you look at the bottom, that's about 30,000 magnification EM from my lab. You can see part of a mast cell at the top with G showing the granule, part of a mast cell at the bottom, again, G showing granule. N is a nerve terminus. The little arrows indicate synaptic vesicles. Well, consider that 50% of the mast cell granules, proteolytic enzymes like meat tenderizers. Of course, they're going to damage the nerve ending. And if you look at the left-hand side from a recent review that we published, you can see that there might be triggers coming, let's say, from the skin in this particular instance, either directly to the mast cell or through blood vessels. The mast cell stimulates the sensory nerves by releasing all kinds of molecules. Clearly, patients are stressed, especially if they're itching like crazy or for whatever other reason. They release corticotropin and releasing hormone, about which I will tell you. That resets the reactivity of the mast cells. The mast cells release molecules activating microglia in the brain, and now you've got a mess in your hands. Because you cannot actually go trying to block 20, 50 different mediators. You've got to try to actually contain the mast cell. And we showed that by many publications. In one, we actually edit technetium gluceptate in the tail vein of a mouse. The technetium gluceptate should not escape the circulation. We put the mouse into a plexiglass immobilizer for 30 minutes. And if you look at the right-hand side, look at the big bar, most of the technetium gluceptate left the circulation and went into the brain, meaning the red circle. But when we did this experiment in mice that did not have any mast cells, called WLW mice, nothing happened. So we know that stress activates the mast cells and resets the reactivity. And we know that actually from experience. We get patients that might have had, let's say, a serious car accident, death in the family, they got sick with COVID, and all of a sudden they're like allergic to life. These are the kind of patients that come to our clinic. And we showed later that the first hormone released under stress, corticotropin releasing hormone, actually not only stimulates the mast cells without degranulation, but resets the reactivity. And then we're using antibodies fluorescent against the CRH1 type of receptor. As you can see at the right-hand side, we showed that human culture mast cells light up like a light bulb. No one had ever thought that a process via stress can have actually molecular applications in how to stimulate the mast cells. And to make things worse, if you look at the middle publication from PNAS, the substance P that is released from mast cells makes the mast cells grow receptors for CRH and vice versa. And if you look at the bottom again from PNAS, and then we also published in PLOS ONE, neurotensin, another peptide, can do the same thing, makes the muscles grow receptors for CR8s and vice versa. So I'm a tremendous proponent of somehow calming our patients, no matter how we can do it, because stress does get the processes going. And back in 1990, I published a review where I called the mast cells the immune gate to the brain. Why? If you look at the left-hand side caricature, you see that the violet are the endothelial cells, the blue are the pericytes that make up the blood-brain barrier, and then you've got sort of podocytes from astrocytes sort of tightening that. But you see a mast cell showing at 12 o'clock. Now, if you look at the right-hand side, about 20,000 diameter magnification from my old lab, you can see an erythrocyte in the lumen of a blood vessel from the brain. Then you see the E is the endothelial cell, P is the pericyte. And then from 12 o'clock to about 7 o'clock, you see a mast cell hugging basically the blood-brain barrier. If the mast cells are activated, they will disrupt the blood-brain barrier. And the mast cells are found uniquely, if you look at the right-hand side, in the hypothalamus and the amygdala. They regulate homeostasis and behavior, and they're found in the meninges. And we'll talk about the meninges shortly. So in the course of our research, and after I joined basically the Institute, we looked to see if there's any association or similarities between COVID-19 and ME/CSF. And we published a paper at the top in the Annals of Clinical Translational Neurology, where we actually found that there were only 8 genes that were actually common to both conditions. And if you look at the left-hand side, in terms of category, most of them were involved with inflammation, T-cell activation, or TLR receptors. So it's music to our ears because that's what we've been discussing so far. And you see at the bottom, causes and symptoms persistent in long COVID and ME/CSF. And in fact, many of our colleagues at the Institute are trying to find out similarities and how to address them. Well, what is the evidence that the mast cells may be involved in COVID? Well, you see at the left-hand side some reviews that I published, but at the right-hand side, you see mast cells in alveolar septa of patients that died with COVID. And the idea was that mast cells are plentiful in the lungs. They release pro-inflammatory molecules. Those can cause edema, inflammation, microemboli. And this is what we call cytokine storms that kill the patients. And at the left-hand side, you see that the same type of thing might be going on, maybe to a limited extent, in long COVID. And 2 papers that were sort of amazing. If you look at the bottom center, mast cells promote viral entry by binding to this enzyme chymase. And at the right-hand side, that histamine potentiates spike protein entry into endothelial cells. And we'll talk about that as well. So is there any direct evidence that mast cells activate actually are activated by COVID-19? You see at the top left, signatures of mast cell activation. They did experiments both in animals, and you can see the animals of the blue panel. The red arrows indicate the mast cells in the lungs of mice that were infected were degranulating. And at the right 2 graphs, in the mice and in humans at the bottom, they measure the enzyme chymase. And in fact, chymase was released. And depending on the severity, the more severe the patient, the more chymase was released. There was also a paper you can see on the right-hand side where they measured both chymase and tryptase in the serum of patients. Both chymase and tryptase were actually high. And at the left-hand side at the bottom, they actually used human culture mast cells, and they used either the pseudovirus or the receptor binding domain of the coronavirus, and they showed that they could actually release both tryptase and chymase. Now, we actually did studies of the same. At the left-hand side, you see a paper published in the New England Journal of Medicine where we used human culture mast cells. We stimulate them with spike protein, either in the presence of absence of either substance P or interleukin-33, and the combination where we gave us the most release of interleukin-1 beta. And then we looked to see what receptors were actually involved in this stimulation. And we pre-treated the human culture mast cells with antibodies blocking either the ACE2 receptor to which the virus binds or the toll-like receptors. And we showed that actually the stimulation was through TLR4. So we might have to worry about inflammation through TLR receptors and not necessarily just the binding and infection of the cells. Well, since the last few years, we all know there is tons of papers showing that there are neurologic symptoms in COVID. And you see some of these papers. And we published a paper with one of our students, basically at the college, about long COVID neuropathy. Now, most importantly, we have actually long COVID, and the long COVID symptoms are somewhat unique because, as you can see at the red box, we have brain fog, confusion, dizziness, dysautonomia, fatigue, many symptoms that mimic ME/CSF. And you can see at the left-hand side in some publications to this extent. And one publication at the bottom where they're talking about cognitive symptoms and ME/CSF as well. So again, we were trying to figure out if there are commonalities. And the center panel summarizes the pathological processes that have been so far been presented. There's blood-brain barrier disruption, there's perivascular inflammation, there's microglia activation, there's neuronal damage. And as you can see, there are publications supporting each one of these contentions showing on this slide. And in fact, in the New England Journal of Medicine, at the upper left, you see some of the early publications that there was actually microvascular injury in the brains of patients. And since then, there are other papers, including one review of mine with my colleagues from Greece, talking about the role of platelet activating factor in microthrombosis and how the spike protein can in fact damage the endothelial cells directly. But where is the spike coming from? Okay, so people actually get sick, and then months later, they have symptoms that linger on up to 3 years, and presumably that's not associated with the severity of the original infection. So there are many papers recently talking about persistent reservoirs in chronic COVID. And one particularly shown at the right-hand side specifically indicated that such a reservoir might be the meninges. And I'm reminding you again that the mast cells are plentiful in the meninges. In fact, we have more mast cells in the meninges than we have in our skin. So we made the hypothesis months ago that in fact the spike protein might be involved in long COVID. And in this caricature shown at the right-hand side from a paper that will appear shortly in Frontiers of Neurology with a colleague from Greece, we showed that the mast cells are found in the meninges that can be activated directly by the spike protein, and they will then activate the microglia, or the spike protein can enter the brain and then activate the microglia directly, and then you have kind of a human continuous reaction. In fact, a paper published about 2 years ago, shown in the middle panel left, indicated that at least the lung mast cells might act as a reservoir for the coronavirus or the spike protein. And I dug back into the literature and I found this report in 2007 where human tissue mast cells were shown to be inducible reservoir for HIV. In fact, in our laboratories, we're trying to find out if after activation, the mast cells might be taking up spike protein. And that's because the mast cells can stimulate the microglia, microglia can stimulate the mast cells, and you can see publications supporting the fact, including one at the upper right-hand side, where activated brain mast cells can contribute to, especially in this case, postoperative cognitive dysfunction. So from our recent publications, we focused on something called matrix metalloproteinase 9, and you see at the top a review how that is important in learning, memory, and neuropsychiatric disorders, because MMP9 cleaves basically the scaffolding both in the brain and outside the brain. It's a tissue disruptor. And we actually published a paper where we showed MMP9 was high in long COVID patients, as you can see. And when we stimulated human culture, actually mast cells, we showed that there was release of MMP9 due to the spike protein. We then went ahead, and these are very recent papers. You can see also MMP9 in translation, how more and more it is considered to be a target for possible therapy. This paper was just accepted in Frontiers in Immunology, and we showed that, again, MMP9 was very high in ME/CSF patients, along with an interleukin-11, which has been associated with lack of longevity, but we'll talk about it some other time. And here we show that the EBV, recombinant EBV protein, could stimulate release of MMP9. We went ahead to show that the SARS-CoV protein can stimulate actually culture microglia. And again, this effect was through TLR4. And you can see on the right-hand side, TLR4 being involved also potentially to increase the expression of ACE2 receptors on the target cells. And we also were very interested in the fact that many papers seem to indicate that EBV virus, which is associated with mononucleosis, may be involved in ME/CSF. And we have a paper almost accepted, actually, in scientific reports, where we show that among many other mediators, mast cells are triggered by the EBV virus to release interleukin-8, both the mast cells and the microglia. So, winding down, what can we do to address mast cell activation disorders? Well, obviously, we should avoid at least any trigger that might be associated. On the left-hand side, you see a number of such triggers. Clearly, if it is histamine intolerance, we can use diamine supplements, diamine oxidase supplements. But as you can see, be cognizant of the fact that even though the amount pretty much says it's about 4.2 milligrams, the activity varies from 10,000 to 20,000 to 18,000 to 1 million, actually. So go figure which one is actually purified enough. But keep in mind, diamine oxidase is an enzyme. One passage through the stomach will be pretty much inactivated by the acidity in the stomach. So if you were to choose one, I would choose for anything that says gastro-resistant or enteric coated. Of course, we use antihistamines, and there are many different antihistamines. There are also drugs that have antihistaminic activity, such as tricyclic antidepressants or phenothiazines. I'd like to stress at least one type of antihistamine called rupatadine, because as you can see at the right-hand side, we published 2 papers many years ago that rupatadine, which is an excellent histamine 1 blocker, also blocked mast cells to different triggers. Another paper compared rupatadine to desloratadine in terms of inhibition, in terms of activation by PUF, and they showed only rupatadine did that. And at the bottom, there is a double-blind placebo-controlled study by my late good friend and colleague, Dr. Maurer, who showed that actually rupatadine improved the quality of life in mastocytosis patients. Now, you might ask, is there any mast cell blocker? Well, the only mast cell blocker sold by prescription is cromolyn, as you can see at the upper hand side corner. But cromolyn is absorbed less than 5% from the gut. The body gets used to it very quickly. And as we increase the concentration, it causes actually diarrhea and loss of hair. And there are publications that are very worrisome as well. Publication by Dr. Church in England, where he had to use very high amounts, as you can see in the red box, before he could inhibit. He also showed that if we push the concentration, it started causing histamine release. And my good friend and colleague, Dr. Gali, who is chief of pathology at Stanford, published papers that it doesn't even work in mice. We have been studying a class of molecules called flavonoids for years. And at the upper left, I show you a review from Pharmacological Reviews from 2000. That's the structure of luteolin. Let me just review quickly by saying it's antioxidant, it's anti-inflammatory, blocks mast cells, blocks microglia, it's neuroprotective, it's a weak metal chelator, increases memory. Well, you can't go wrong having so many benefits. And in fact, a number of papers have shown, as you can see in the middle panels, that luteolin can be both a mast cell inhibitor and anti-inflammatory agent. Well, about 2 years ago, we bit the bullet and we used culture human mast cells and we compared head-to-head cromolyn to luteolin, pre-treatment 10 minutes, and then stimulated either with Ig anti-Ig, like an allergic trigger, or interleukin-33, a non-allergic trigger, and we measure different mediators, as you can see. The first bar is control, the second bar is stimulation, third bar is inhibition by luteolin, fourth bar is inhibition by cromolyn, and as you can see, in every instance, luteolin was much better inhibitor than cromolyn. In fact, if you look at the upper right-hand side of this, cromolyn stimulated actually interleukin-1 beta rather than inhibiting it. And we went ahead to show the pre-treatment of microglia with luteolin when they were stimulated with ochratoxin A, which we don't have enough time to go into, inhibited actual release of MMP9, and a new molecule called methlut was even better inhibitor. And you can see in the right-hand side papers showing that luteolin can actually work either in models of autism or models of LPS-induced sepsis. Now, this is a busy slide, but I want to very quickly review something that is very confusing to everybody I ever talked to. Everybody says polyphenols are good for you. You go out there and you buy polyphenols, biophenols, this, that, the other thing. And we think that the more phenolic, the better things would be. So at the left-hand side, I show various phenolic compounds and how many phenols they have. A phenol is a hydroxy group bound to a benzene ring. And you can see luteolin has 4 phenols, so it's not at the beginning, it's not at the end. And then I calculated basically what we call IC50, how much do you need to inhibit mast cell secretion by 50%. And you can see luteolin is less than 1 microgram. And even though many other molecules are used to inhibit mast cells, the concentrations you need are actually sometimes 100 times more than we need for luteolin. So don't necessarily think that any polyphenolic compound will do the job that you want it to do. So how does luteolin actually work? Well, first of all, you can see in this caricature that it can block the Fc epsilon receptor for Ig, it can block the MRG receptor, it can block TLR receptors, and it can block the ST2 receptor for interleukin 33. It also blocks mTOR, mammalian target rapamycin, NF-kappa beta, and the inflammasome. Again, I don't know of any molecule that can block as many targets as that. And because flavonoids, including luteolin, are absorbed less than 10% from the gut, clearly making them liposomal would be best. And I helped actually formulate a number of supplements, as it was mentioned in the introduction, where we use olive pomace oil to make basically liposomes that include a luteolin. And in fact, there have been many reviews about luteolin being used in brain fog and chemo fog associated with chemotherapy of cancer. Now, I want to show you this at the right-hand side because you might not be aware of it. If a company wants to export a supplement, a dietary supplement, they can go to the FDA voluntarily. And if the company is FDA registered, GMP certified, the content is reviewed by an independent source, in this case, Eurofin, and there's at least one open label study, then they give you this certificate of free sale. And as you can see, it says FDA gives you, the golden seal there. It's renewable every 2 years. So we can raise basically the bar for the kind of supplements that we should actually be using for our patients. And since then, we've identified a new molecule, tetramethoxyluteolin, where there are no basically hydroxy groups or methyl groups. The beauty of it is, as you can see at the right-hand side at the top and at the bottom, it blocks actually both mast cells at the top and at the bottom microglia better than cromolyn and better than luteolin. And we published a number of papers, including methoxyluteolin inhibiting substance P stimulation of mast cells and how it blocks mTOR activation. And because of the lack of hydroxy groups, methoxyluteolin does not have color because otherwise the flavonoids are yellow. And you can see in the middle right panel a paper where it was used, methoxyluteolin was used in a skin lotion with very beneficial effects and improvement in eczema. So I think I have only 2 slides left. If we look at how do we treat mast cell activation disorders, well, we start with antihistamines. You can go up to 4 times, and you can include rupatinib because it blocks PAF and eosinophils. Then you can actually add histamine 2 blockers such as famotidine. We can add flavonoids like luteolin and quercetin, especially if they're formulated in liposomal form. If there's a lot of GI problems or there's a lot of bronchoconstriction, you can add the montelukast, which blocks leukotrienes, or you can use cromolyn, even though it doesn't get absorbed very well. If you have a lot of asthma associated with eosinophils, or itching associated with eosinophils, now there are new drugs such as dumpilumab, mepolizumab, etc., that target either IL-4 or IL-5. And eventually, if you have systemic mastocytosis, many drugs exist that block the tyrosine kinase, which is literally c-KIT. But as you can see in the kind of recent review showing at the right, even though we have many such drugs, they induce apoptosis of mast cells, but they do not inhibit activation of mast cells. So here's a review about mast cell activation beyond histamine and tryptase. The summary is that mast cells are ubiquitous in the body. They can be triggered by multiple chemicals and other triggers. They can release, hundreds of molecules. There's no real drug that blocks the mast cells. Some flavonoids, especially liposomal form, do it. And this DAISY is meant to show that the color petals are where we absolutely know mast cells are involved, but the white petals are conditions where we suspect mast cells are involved, and we should definitely try to address them. So what I will show you here, it was part of the supplemental. This is the last slide of the review in the New England Journal of Medicine. It's what you see there, and I will activate it shortly, is one mast cell between two cover slips stimulated by perfusing substance P, and you will see what happens in real time. And what you see in the middle here is the nucleus, and the bumps that you see are actually secretory granules. It's never been published before except in the New England Journal of Medicine supplemental. See what happens. So this is real time as substance P perfuses. So seconds, look what happens in seconds. So whenever we deal with mast cell activation problems, we don't have enough time. And antihistamines will block only one, as I said, mediator. So the best approach would be to try to kind of block the mast cells from releasing most of the molecules and then have the antihistamines or what have you as a last resort. And we're struggling very much to identify how to actually block the mast cells, obviously better than just using luteolin. Thank you very much for listening.
Thank you so much. We'll open the floor now for any questions, and those of you who may need to jump off, please feel free to complete the evaluation. And for those who still have time or have questions, we'll go ahead and stay on a few minutes to answer those. Okay, so it appears that there are no questions. So if there are no questions,
Goodness.
Please. Oh,
there is one from Dr. Mitchell.
Yes, if I could ask Dr. Theoharides, by the way, amazing lecture. I mean, beyond amazing. But I do have one question that I always think about. You know, again, what I see in clinical practice in my allergy and immunology practice, I'm always fascinated by how fast a reaction can happen. You know, a peanut allergy, a smell, obviously with the patients that I see with toxic mold. My question to you is, if there's an answer to this, these tissues are, you know, the mast cells are localized, whether it's in the nose when a patient first breathes in something or in their stomach. How does it happen within minutes? Is it through the nervous system? I mean, how does that, you know, because again, you can understand when something inflames an area, it'll be local.
Sure,
but these patients, let's say, obviously anaphylaxis or some kind of toxic mold thing, it's
you're absolutely
widespread.
right. Absolutely right. In fact, many of our patients say they're triggered by smells, whether it's smell associated to peanuts or smell associated with Clorox or benzene or whatever.
Right.
I've tried 5 times to get funded for the following. We know the olfactory receptors. So I've asked for funding to see if the mast cells actually express olfactory receptors, in which case it will be triggered immediately. And the government's never actually funded me. They turned me down every time. They thought it was crazy to think mast cells might have that. I think I'm going to just beg for donations to do this, because if this is the case, it will change our thinking about the mast cells are activated, at least in the nose. But I don't know the answer.
But my question is, though, do you think, though, it's the mediator release, or is it through, because you mentioned, you show how the mast cells are draped around vessels,
right?
You know, because they don't circulate, or next to nervous system. Does the nervous, I mean, like, what's the mechanism that gets it so generalized reaction?
Correct. Without necessarily saying that what I'm about to say is the beginning and the end, but, you know, stress, you know, is both physiological and psychological stress. So if someone goes through duty free and all of a sudden is hit by, you know, 100 different smells, they're going to trigger your brain, and your brain will release corticotropic releasing hormone and substance P and angiotensin all over the body, and they will basically trigger mast cells. And there's antidromic and orthodromic stimulation of the nerves as well. But in all honesty, Dr. Mitchell, you're absolutely right. I've been studying mast cells for 40 years, and I'm so absolutely sad I cannot regulate the mast cells. You know, it's just amazing. We're dealing with complicated, you know, conditions. And here we have one single cell that participates in so many conditions.
Okay.
We cannot regulate it.
And then why does epinephrine work so well?
That's different, though. So part of the symptoms of the mast cells are bronchoconstriction due to leukotrienes and histamine and VEGF that causes blood pressure. Epinephrine does not block mast cells. Epinephrine will open up your bronchi and basically dilate, constrict your blood vessels so you don't drop your blood pressure. So basically,
so it doesn't. You don't think it doesn't like kind of squeeze the mast cells?
It doesn't. It doesn't. There were some reports from many years ago where they studied lung mast cells, and they showed that maybe epinephrine did something in the lung mast cells, but some of those were refuted afterwards. I'm not saying that epinephrine might not have some effect, but the primary effect in clinical medicine is to dilate, prevent constriction of the bronchi and prevent dilation of the blood vessels, so you can breathe and you don't basically fall and hit your head.
Thank you. Amazing,
yep.
Amazing lecture.
Thank you all again for listening.
All right, so any other questions? If not, we wish you all a wonderful day, and the materials will be available on the faculty portal.
Thank you.
Thanks.
Okay.
This is